Hello, everybody. Welcome back to the third session of the microbial environments theme day. So today we're really excited to bring to you a talk by Dr. Erica Hartmann. Some background on Erica is that she's an environmental microbiologist interested in interaction between anthropogenic chemicals and microorganisms, as well as bio-inspired mechanisms for controlling microbial communities. Her career began at the Johns Hopkins Bloomberg School of Public Health, where she worked on mass spec-based methods for detecting microbial enzymes necessary for bioremediation. She then moved to Arizona State University, where she was the first graduate of the interdisciplinary biological design PhD program. She then moved to France on a Fulbright study, studying microbes that degrade carcinogenic pollutants at the Commission for Atomic Energy. She then began leading studies on antimicrobial chemicals and microbes and indoor dust at Biobe in Oregon. And she's currently contributing that work as a well-developed non-chemical antimicrobials as an assistant professor at Northwestern University. Thanks, Erica, and we're really looking forward to your talk.
Thank you, and I'm so sorry, I need 1 second to step away. I will be right back.
That's fine. That's fine.
So sorry about that.
Erica, not a problem at all. Not a problem at all.
So
good luck.
Thank you. And thank you very much for the invitation to speak today. So I am really interested in microbe-chemical interactions. And so today I'm going to talk about a very specific type of microbe-chemical interaction that involves antimicrobial chemicals and antimicrobial resistance, and specifically in the context of the indoor microbiome. And of course, I'm happy to take questions and discuss during the next hour, but also I invite anyone who wants to continue the conversation to find me either on Twitter or Instagram or both at Erica M. Hartmann. So with that, I think probably I don't need to explain at this point in the day to this particular audience. Hopefully everybody already knows that the built environment, the environment around us, is not sterile, and that we as sort of walking, talking bioreactors are bringing our microbes with us everywhere we go. So of course we have microbes, in our gut, on our skin, in our mouths, etc. And we shed these microbes in our own personal microbial aura everywhere we go. And that includes the environment where we spend 90+% of our time, which is to say the built environment or indoors. And so of course this is very important because, as my colleague Jack Gilbert has previously demonstrated, not only are we shedding our microbes, but we're also picking up microbes from the built environment. And so there's this potential for exchange, and that exchange is potentially very important for lots of different types of health outcomes. So Jack and others have done work showing that it's not just us shedding our microbial auras, but that we're picking up microbes. And this can have implications for things like allergies and asthma potentially, but also what I'm really interested in is the potential for acquisition of antimicrobial resistant pathogens. And this is obviously Jack did this work in a hospital, but it's also been looked at in other contexts. Hospitals are obviously very sensitive environments for this kind of partitioning and sharing. So one of the things that we know already about microbiomes of the built environment is that they are shaped by human activity and a variety of selective pressures. And one of the things that I think is so interesting about the built environment is that we are very uniquely positioned to control the types of selective pressures that microbes that surround us are experiencing. So we already saw some probiotic designs with this principle already in mind, thinking about how can we design with microbes in mind, and how can we use some of this some of our ability to control the materials that we use and the temperature and the humidity and that kind of thing. The kinds of features that we can't really control in external environments like, forests or oceans or things like that. Although we are, of course, having an undue influence on those environments as well. So then if we can shape microbiomes of the built environment based on some of our design choices, what are the types of choices that we might begin to make? So again, we've already seen something about the types of materials that we might use and thinking about probiotic materials. But on the flip side, because we're not, I would say at the moment, that is the exception rather than the norm. So on the flip side, one of the things that we do is we use a lot of antimicrobials. So we introduce a lot of chemicals into our environments, and that comes in various different forms. I'm going to highlight just a few, but I encourage you going forward to take a moment to pause and reflect and really think about the chemical composition of the world around you. So some of the chemicals that I'm really interested in, as I mentioned previously, are antimicrobials. Excuse me. So these might be things like triclosan, which is fairly commonly incorporated, or used to be fairly commonly incorporated in toothpastes, and benzalkonium chloride and other quaternary ammonium compounds, which are currently very widely used in hand soaps and other surface disinfectants. And one of the things that I think is really interesting about these chemicals and our approach to using them is that they're not just found in personal care products, so the things that we use for hygiene, but they're also used in the very materials with which we build our built environments. So you'll find triclosan or quaternary ammonium compounds not just in your toothpaste or your hand soap, but also in the textiles that we use to make our world more comfortable, in the flooring materials that we use, in our surface finishings like our paints that we use on the walls. And so I'm really interested in what happens when we use these antimicrobials in so many different places so sort of indiscriminately. And one of the things that can happen that we know from other environments is that where you have exposure to antimicrobials, you have an increase in resistance. So of course, we're not just using these chemicals and then they disappear. We use these chemicals and we are surrounded by microbes, both our microbes and the rest of the microbes in the built environment. And every time those microbes encounter those antimicrobial chemicals, they have an opportunity to develop resistance. And they have lots of different strategies to develop resistance. So starting from the bottom, we can look at, for example, transformation. And this is a really interesting one to think about in the context of the built environment, because that's really impacting the chemistry that surrounds us, right? So we're starting with one chemical and then there's potentially biotransformation that's happening, which means that actually what we're exposed to isn't what we think we're putting out there. Another strategy, so the one depicted in the middle, is spore formation. So we've already heard a little bit, I think, about an allusion to spore formation as a survival strategy. This is really interesting. This and other lifestyle survival strategies like biofilm formation are interesting reactions to antimicrobials because they also provide protection to a lot of other potential physical stressors that one might encounter in the built environment, such as desiccation or nutrient limitation. So again, this idea that these dry, barren surfaces in the built environment aren't necessarily where microbes want to be living and metabolizing and doing all of that fun stuff. And then another strategy that microbes can use, and this one is particularly problematic, is efflux. And the reason it's particularly problematic is because sometimes this efflux is non-specific. And so the same efflux mechanism that will protect a microbe against, for example, triclosan, may also protect it against clinically relevant antibiotics. And this is a really big deal if that microbe then causes an infection. We're then incapable of treating that infection in a way that we used to be able to handle very easily. So here's the sort of backdrop of what happens in general when microbes encounter antimicrobials. And it's a really, really big challenge because of the sort of global relevance to public health. So I'll say there's a lot of skepticism around projections of antimicrobial resistance. We don't obviously know the exact numbers, but I think everyone basically agrees that antimicrobial resistance is on the rise and it's hugely problematic. Again, as I was saying, because we, over the last 100 years, have been so fortunate to have a fairly, usable arsenal of antibiotics that can treat all of these diseases that used to essentially be fatal. And if we don't do something about antimicrobial resistance, we're going to go back 100, 150 years in time to where a lot of these things that are really not a big deal now can become fatal. So I'll say, the microbiome of the built environment is only one aspect of fighting antimicrobial resistance, and it's only one part of a very, very large puzzle that needs to be solved in order to prevent the sort of doomsday scenario that some people are projecting, and I think that everyone is preparing for. Okay, so in that context, I invite you now to think about the built environment, the environment around you, not as the static, four walls and a ceiling that I think maybe some of us take for granted, but as a dynamic chemical and biological system where we have chemicals that are coming in from indoor sources. So those are going to be our personal care products, but also our building materials, all of the various things that we bring in and all of the various things that we build with, as well as, of course, outdoor sources. So that's going to be things like what's showing up in air. And as we start to think more and more about air quality, I think this is a really important thing to keep in mind. And these chemicals get into the built environment, but they don't stay sort of where they come from. They partition. So some things are going to volatilize into the gas phase. Some things are going to absorb to suspended particles, and sort of regardless of where things initially partition, eventually gravity works and everything winds up in settled dust. And this is important because then dust becomes this sort of repository for all of the chemicals that have been in the air, but also, of course, all of the microbes, because as we've already discussed, there are microbes all around us. And so going forward, again, I would like you to think about not just, oh, I'm surrounded by a building, but I'm surrounded by a physical structure that houses a dynamic chemical and biological system where we have all of these chemicals that are moving around, all of these microbes that are moving around and potentially adapting to the chemical exposures. So if that I just presented is the case, one might ask the question, is resistance in microbiomes of the built environment actually shaped by antimicrobial usage? This is a great hypothesis. Is it actually happening? And since we've been using antimicrobials for many decades now, it's a question that we can start to answer. And we have evidence of this from other scenarios. So obviously antibiotics, we know from studies of the human gut that where we perturb these resident microbial communities in the human gut with antibiotics, we alter the population structure of the gut microbiome, and we can also enrich for antimicrobial resistance. And so is this same phenomenon happening with antimicrobials, some of which may draw some parallels to clinically relevant antibiotics. And we also know from other environments, so specifically wastewater treatment, that where we perturb microbial communities with triclosan, we see a huge increase in antimicrobial resistance. And so this work, for example, took microbial communities from wastewater treatment plants, which are giant bioreactors. And if you just sort of look at the community over time, it's generally fairly stable. And in this case, what they were specifically looking at was MEX-B, which is one of those multi-drug efflux pumps. But if you perturb the system with triclosan, in just over 2 weeks, you see over an order of magnitude increase in the abundance of these multi-drug efflux pumps. So if, in fact, we're using these chemicals and those chemicals are enriching for antimicrobial resistance, particularly in the form of multi-drug efflux, this is probably not a great thing. All right. So one study that I have done to specifically look at the impacts of triclosan on antimicrobial resistance in the built environment, I'll walk through in just a second, but again, a little reminder about triclosan. So it is, at least in the United States, I think generally being phased out of toothpaste. It was one of the antimicrobials that was banned by the FDA a few years ago, but it is still found in a variety of products throughout the world. And interestingly, it is ubiquitous in indoor dust. So there have been a handful of studies that have looked at triclosan and other antimicrobials and other semi-volatile organic chemicals in indoor dust throughout the world. And almost every sample that's ever been collected anywhere contains contains measurable quantities of triclosan, and actually quantities of triclosan that approach what we see in wastewater treatment, which is one of the places where we think that triclosan is particularly problematic. And we know from lab studies that triclosan is linked to antibiotic efflux. So as I mentioned previously, it's also linked to horizontal transfer of mobile antibiotic resistance genes. So just the fact that triclosan is present can stress out microbes, which then prompts them to start sharing mobile genetic elements, some of which contain antibiotic resistance genes, even though those genes may not confer any advantage against triclosan itself. And so to look at whether or not those types of phenomena are taking place in indoor dust, we performed a study where we went around and collected a bunch of dust samples using vacuums, as one does. And we measured triclosan and a handful of other antimicrobials using liquid chromatography tandem mass spectrometry. And this allows us to very specifically identify and quantify the amount of antimicrobials present. We also identified antibiotic resistance genes using shotgun metagenomic sequencing, and we identified antibiotic resistance genes based on their homology to known antibiotic resistance genes. So of course, there are some limitations to this approach, but it's a pretty good place to at least start. And then to complement that, we also did culture work, and we cultured antibiotic resistant bacteria so that we're able to look not just at the genetic material that's there, but also the actual viable microbes. And to cut to the chase, what we saw is that in samples that had higher concentrations of triclosan, we tended to see genera of microbes, and this is from the metagenomic data, that are associated with more frequent or more diverse antimicrobial resistance determinants. So what you're seeing here is a list of the genera that we were able to identify their association with either triclosan or a related antimicrobial triclocarban, and the frequency of annotation of different antimicrobial resistance genes in the pangenome of that organism. So where we see higher concentrations of antimicrobials like triclosan, we see more and more diverse antimicrobial resistance genes. And this is just in the metagenomic data, so that's not necessarily indicative of what's alive and what's surviving in that environment. So we then looked at the culture data, and what we saw here, and this is a little tricky because we can't actually identify all of the thousands of organisms that we were able to culture. But what we did see is in samples where we had, again, higher concentrations of triclosan, we tended to see clusters of organisms that were more likely to be resistant to more antibiotics. So we tested 3 antibiotics, which represent 3 different classes of antimicrobials: clarithromycin, ampicillin, and tetracycline. And where we had higher concentrations of triclosan, we tended to find organisms that were more organisms that were resistant to all 3 of these. So again, kind of concerning. So one might then ask the question, if resistance in microbiomes of the built environment is in fact shaped by antimicrobial usage, where does that resistance come from? So if we're selecting for resistant organisms, where is that resistance coming from? It doesn't just appear out of nowhere, it has to come from somewhere. And one of the things that's really interesting about the built environment is that it's sort of a meeting ground for microbes from a bunch of different places. So as I already mentioned, we as humans are bringing our microbes in, and we're shedding those microbes all around us, everything we touch, everywhere we go. We have our microbial cloud that's being deposited around us. But we also have microbes that are coming in from external environments. So the things that are coming in, that you're tracking in on your shoes, or the things that are coming in from outdoor air. And so we have this opportunity for human-associated microbes and environmental microbes to get together and experience some interesting conditions. And then, of course, as we go through, we can then pick those back up. And one thing that can happen when these microbes are together experiencing these unique conditions, especially ones that are particularly harsh, is that it might favor the transfer of antimicrobial resistance genes. And so going back to that idea of horizontal gene transfer, and this idea that environmental microbiomes are notorious for having very diverse antimicrobial resistance genes, and human-associated microbiomes are notorious for having highly abundant antimicrobial resistance genes. And so when you bring these things together, what we may find is just that we're increasing the overall abundance of antimicrobial resistance around us and also the diversity. So challenging any existing or new antibiotics that we may come up with. So one thing that we found in that previous study I mentioned about dust is that different samples had different proportions of human-sourced microbes. So within the built environment, if you have different ventilation strategies or different occupancy behaviors, you can see more or less human-associated microbes compared to environmental or other sources. And one of the things that we noticed, so we looked at the presence of operable windows or exterior doors, so a direct sort of inlet for outdoor air. And where there were no operable windows or exterior doors, there was a slightly higher proportion of human-sourced microbes. So we're sort of enriching for the human-associated microbes around us. And somewhat interestingly, where we had daily cleaning with what we call chemical cleaning, so this isn't like dusting and sweeping, but actually using chemical cleaning products, we also saw a higher proportion of human-sourced microbes. And we don't really know why that is, but we think it's interesting. And we think it's interesting for several different reasons. One is that if we are concerned about pathogens, typically the pathogens we're concerned about are those human-associated microbes. But also if we're concerned about antimicrobial resistance, the introduction of new antimicrobial resistance determinants is likely going to be coming from environmental microbes. So what then do we do about this environmental versus human-sourced microbes problem? So another environment in which we wanted to look at this question of where are microbes coming from and where are their resistance determinants coming from is toothbrushes. So toothbrushes are a really unique built environment sort of niche. One reason that makes them very unique is that they have frequent, hopefully daily or twice daily, direct contact with humans. So you might say, oh, I'm not that concerned about the microbes on my walls because I don't actually rub up against my walls or whatever. But you hopefully are, putting your toothbrush in your mouth at least twice a day. So we have a direct route of transmission for human, oral, and skin-associated microbes. We also have, very importantly, periodic wetting. So I think it's already been mentioned that the presence of water is critically important for microbes' ability to survive, especially in the very harsh conditions where we might have a variety of temperatures, varying availability of nutrients, and of course the presence of various cleaning products. So your toothpaste, which in addition to potentially having triclosan, definitely has a detergent in it. So we did a study where we had people mail in their used toothbrushes. I'm very grateful to all of the participants who mailed us their toothbrushes. And we compared using, again, shotgun metagenomic sequencing, the diversity of microbes on toothbrushes to the diversity that's observed in the human mouth through the Human Microbiome Project. And what we saw is that the human mouth tends to be more diverse than toothbrushes when it just comes to microbial taxa. And some of the, there we go. Some of the factors that influence diversity on toothbrushes are some behavioral factors like how often you brush your teeth, more than twice a day versus once a day, or where you store your toothbrush. So whether you're keeping it in an enclosed environment like a cabinet or a drawer, or whether you're leaving it out on the vanity in your bathroom. So those have somewhat weak but noticeable impacts on the diversity of the toothbrush microbiome. But one of the things that we also noticed then is not just that toothbrushes are less diverse, but they have more diverse antibiotic resistance genes. So we thought this was really interesting, that we're having a decrease in overall taxonomic diversity, but an increase in diversity of antibiotic resistance genes. And so what's driving that? Where are those antibiotic resistance genes coming from? So we can go back to our data set and look at the antimicrobial resistance genes that are present in the human mouth as determined by the Human Microbiome Project, and those that we find on toothbrushes. And so what you see here is a list of antimicrobial resistance genes, and the orange side are the ones that are detected in the Human Microbiome Project, so the human mouth, and what's in red is what's detected in a toothbrush. And we see that there are a few antimicrobial resistance genes that are highly prevalent. So they're detected in a lot of Human Microbiome Project samples, but more antimicrobial resistance genes that are detected on toothbrushes, and especially at lower frequencies. So we're not necessarily seeing just one antimicrobial resistance gene that's everywhere in all toothbrushes. We're seeing a lot of different things that encode for a lot of different classes of antimicrobial resistance. So high diversity, but low frequency. And we suspected that that high diversity but low frequency was coming from sporadic inputs from environmental microbes. And so sort of to corroborate that hypothesis, we looked at the fraction of human-derived taxa, so the proportion of microbes that are associated with the human mouth as opposed to other environments. And where we saw less human-associated microbes, we saw more antimicrobial resistance genes, so higher abundance in this case. So that sort of corroborates our hypothesis that where we have a lot of diverse antimicrobial resistance in the built environment, or at least on toothbrushes, that diversity is really coming from environmental microbes. Okay, so we have this dynamic environment where we have lots of chemicals that are coming from different places. We have lots of microbes that are coming from different places. And I've showed you correlative studies that looked at the association between the presence of antimicrobials and the presence of antimicrobial resistance. But are we actually actively selecting for resistance in the built environment? Because if so, that's obviously highly problematic. So to answer this question, we started to look at benzalkonium chloride, which is the replacement for triclosan in antibacterial hand soap in the United States, and an extremely widely used disinfectant worldwide. In fact, during the COVID-19 pandemic, the demand for benzalkonium chloride and quaternary ammonium compounds exceeded the production capacity. So the entire industry basically hit the ceiling. And this is not only because of antibacterial hand soaps, but also because this is the active ingredient in a lot of those surface wipes, so disinfecting wipes, and also a lot of other surface cleaners. Interestingly, benzalkonium chloride is also the active ingredient in, as far as I'm aware, the world's first biocidal paint. So a lot of wall paints contain an antimicrobial to prevent mold from growing on the paint while it's in the can. But this paint actually has been proven to kill MRSA and E. coli and whatever else. So as I mentioned, benzalkonium chloride is a replacement for triclosan in soap, but it is known to have a few weaknesses. One is that it's known to be ineffective against spores, and this is particularly problematic in healthcare environments if one of the things you're concerned about is C. diff, which is notoriously spore forming. It's also linked, much like triclosan, to antibiotic transformation and efflux. So microbes that are exposed to benzalkonium chloride are perhaps more likely to develop cross-resistance to, again, clinically relevant antibiotics that we need to save lives and cure infections. So we did a study where actually using those microbes that we were able to culture from indoor dust, we created a mock indoor microbiome community, so something that's representative of the types of microbes that are actually surviving in built environments. And we exposed these mock communities to 3 different types of paint. So as I mentioned, there's this microbicidal paint, which contains benzalkonium chloride and has been demonstrated to kill 99.9% of 5 different pathogens under lab conditions using standardized testing. We also used a conventional mold-resistant paint. So this is just a standard latex paint, which, as I mentioned, many paints contain an antimicrobial. They won't necessarily tell you what the antimicrobial is, but it's really there not to protect your health, but to protect the longevity of the paint while it's in the can. And then we were also curious, in the interest of thinking about new sustainable, not new, greener, more sustainable materials, actually very old materials. We used an all-natural clay paint to see, how the surface chemistry might change if we were using something that's just a very natural, non-synthetic product. And what we saw, somewhat unsurprisingly, is that when we put this mock community on these painted samples, that everything died. Right? It's a dry, barren surface. We didn't give them a lot of water. We didn't give them anything to eat. Everything died pretty quickly. Not everything though. We saw several log reduction, but not everything died. Some things were able to persist on these surfaces. And we measured this using viability quantitative PCR, which is an imperfect method, but nevertheless, we know that even if we're seeing 3 or 4 or even 5 log reduction, we're not fully getting rid of all viable microbes on that surface. But what was really interesting is that even though most things are dying, some things are surviving, and who is surviving is actually impacted by the local conditions. So we had 3 different relative humidity conditions, 0, 20, and 100%. And then we had these different surface conditions. So on the top, you're looking at the clay paints, and on the bottom, you're looking at the regular synthetic paint. And even though these samples all started with the same general microbial community, we see very different community compositions within the surviving community. And this is still true for the biocidal paint. So even though it contains this antimicrobial, these quaternary ammonium compounds, we still see some surviving microbial community. And within that community, we're definitely enriching for, in this case, it's a Bacillus timonensis. And of course, if you're following along and you're a Bacillus aficionado, you know that one of the hallmarks of Bacillus is that they form spores. And I already mentioned that one of the blind spots of benzalkonium chloride is that it's ineffective against spores. So we hypothesized that one reason that the Bacillus were surviving so well on these biocidal surfaces is that it was forming spores. And so we collected some samples and did electron microscopy, and we saw some spores. So on the top, I'm showing these are control samples for vegetative and sporulated Bacillus timonensis. And then on the bottom are samples that we collected from our surfaces. And what's interesting is that we see both vegetative and sporulated forms of the Bacillus. And so we suspect that, yes, spore formation is part of the arsenal of tools that Bacillus use to survive within the built environment, but that there may be other things that the Bacillus is doing as well that allows it to survive for prolonged periods of time, and especially in the presence of quaternary ammonium compounds. Another type of antimicrobial, so we've talked about now triclosan and benzalkonium chloride. I think those are very interesting. Another one that sort of, I would say, comes and goes in popularity is the use of metals, and especially right now, metal nanoparticles. And especially metal nanoparticles are used a lot in textiles. So while I was on the subject of the Toothbrush microbiome project, I came across these antimicrobial toothbrushes. So not just the antimicrobial in the toothpaste, but you can actually buy toothbrushes that have metal nanoparticles embedded in the bristles of the brush itself. So, of course, not surprisingly, the exposure to metal nanoparticles or metals in general is also linked, much like with benzalkonium chloride and triclosan, to horizontal transfer of mobile antibiotic resistance genes. So just a general stress tolerance mechanism. Bacteria get stressed, they start sharing genes. Also, exposure to metals may induce efflux. So without even having a change in the genetic determinants of antimicrobials, you make them temporarily more tolerant by inducing efflux. And they may favor co-selection of metal and antimicrobial resistance genetic determinants, because some of these metal resistance genes and antibiotic resistance genes are co-located on mobile genetic elements. So much like with triclosan and benzalkonium chloride, I had some concerns about our use of metal nanoparticles as antimicrobials, in this case, particularly on toothbrushes. So this time we went out and bought some new toothbrushes. And the first thing that we did is try to look at where these metal nanoparticles are. And so these are, again, electron micrographs where what you're looking at is individual toothbrush bristles, both the side and a sort of transect. And all of the shiny spots are metal nanoparticles. So you can see that some of the nanoparticles are on the surface, but a lot of them are actually in the interior of the bristle. And I think this has to do with the manufacturing process, where they actually mix the polymer and the nanoparticles and then extrude them together. And I don't think I'm based on our data, I am fairly certain that the nanoparticles at the interior of the bristles are basically doing nothing. There's no way for the microbes on the surface to interact with the nanoparticles deep within the material itself. So that's the first issue. The second issue that we saw is we actually, we built a little instrument to reproducibly brush with these toothbrushes. And what we saw is that just due to mechanical abrasion over time, the nanoparticles on the surface would fall out and leave these divots. So you can sort of see there are these little pocks that form in the sides of the bristles. And this is important because the more sort of topology there is on the surface, the easier it is for microbes to attach to that surface. So on that subject, we also took these bristles and exposed microbial cultures to them. In this case, we're using Staphylococcus epidermidis and Rathia dentocariosa. These are both organisms that we were able to culture from our used toothbrushes. And what we saw, these are just the antimicrobial toothbrushes that I'm showing here, that the surface topology did help microbes to adhere to those surfaces. So we saw more microbes growing on the surfaces of the antimicrobial toothbrushes. When the nanoparticles were present, they did end up killing more of the microbes, and we did that using cyto live dead staining. And when they obviously weren't present, they killed fewer of the microbes. Seems obvious. And this is sort of not a super conclusive study. We only had these two organisms, but within these two organisms, we could look at the distribution of both metal resistance determinants and antimicrobial resistance determinants and see where those were co-localized. And again, it sort of suggests that where we're selecting for things that are resistant or tolerant to metals, we're also selecting for things that are resistant or tolerant to antimicrobials. Okay, so I think that's kind of a lot. So just to summarize, what we sort of know already about antimicrobial resistance in the indoor microbiome, we know that it correlates, that antimicrobial resistance correlates with the presence of antimicrobials. We know that antimicrobial resistance is more diverse where there are more environmental microbes. And so really thinking about where our microbes are coming from and what they're bringing with them. And we know that antimicrobial resistance is selected for by antimicrobial products, so those antimicrobial paints or toothbrush bristles. And so with that in mind, when we are going forward and thinking about how do we design our built environments, we should really think about not just the microbiology and the sort of biodiversity aspect, but really think about the chemistry, the microchemical interactions, and particularly the evolutionary aspects of we are in the middle of this giant project where we're potentially experimentally selecting for antimicrobial resistance, which of course can have catastrophic effects for human health long term. So my overarching research really continues to probe the answers to this question of how microbe-chemical interactions affect the indoor exposome, and really thinking about how our choices in the products that we buy and the things that we use within our built environment are impacting both the microbiology and the chemistries that we're exposed to, and how overall that has short and long-term impacts on human health. And a lot of times I'll say, I'll preempt one question, which is, okay, so what do I do? And so my request to everyone who has watched this talk is that going forward, you really think about what types of chemicals you're using in your built environment, what kinds of personal care products, obviously, but also thinking about the upholstery on your couch or your carpet or your paint on your walls, and think, do I really need this to be antimicrobial? Is this really serving a purpose, or is that a marketing gimmick? Because in a lot of cases, so there are obviously some cases like hospitals where we have very critical needs for, sanitary environments, food prep, that kind of thing. But in a lot of cases, and as we've seen previously, there's a drive to promote microbial diversity and also to retain some of that sort of biomass that's around us that may have a protective effect on our health long term. And so to really think, again, do I need this antimicrobial? Even in the absence of some sort of probiotic approach? Can we just step away from the antibiotic approach? All right. So with that, obviously, I did very little of all of the work that I just presented. So I owe an enormous debt of gratitude to my entire team, most of whom are unfortunately not pictured here for reasons. We haven't taken a group photo in a couple of years. But I have some really fabulous trainees and some really fabulous collaborators who have helped with all of this work, and a lot of generous funding organizations. And thank you very much for your attention, and again for the opportunity to speak here, and I'll take any questions.
Thanks, Eric. That's a really lovely talk.
Thank you.
Yeah, I mean, I do have some questions while some are filtered in through YouTube. So regarding the benzalkonium chloride and its inclusion in paint, do you have an idea how quickly the antimicrobial is leached out of the paint? And, because are you seeing initial tolerance, stress, and then accrual of antimicrobial resistance genes, or do you think it's just a straight open system?
So I have a couple of answers to that question. So in the studies that we did, we just freshly painted the surface and then put microbes directly on the surface and left them for up to a week. And even within that week, we already saw an enrichment within the microbial community for tolerance. And that's due to direct contact on the surface. With regard to leaching out of the surface, I think that's really, really interesting because, of course, materials degrade over time. And so there's a possibility for that benzalkonium chloride to actually be leaving the paint and then sort of finding its way around the room in dust, as I mentioned previously. And we haven't done that study, and part of the reason for that is that the analytical chemistry is quite challenging because benzalkonium chloride isn't just one chemical, it's a family of chemicals, which are very, very sticky. But we're super interested in it, and we're sort of working on that now.
That's great. I mean, I just wondered if you'll see like an adaptation, and then if it's slow enough, you may actually see complete resistance through a chromosomal change, rather than being associated with. I mean, and to note that and see would be amazing.
Yeah.
I mean, Jack's posted it in the chat as well. Can antimicrobials in these materials alter the fungal bacterial community balance?
I think that's a great question. So we only used bacteria in those experiments, mostly because I have very little experience in mycology. So any mycologists out there who want to collaborate, please, yes. So when we did our enrichments from built environment dust, we were really selecting for bacteria. We actually used a fungicide so that we weren't getting huge fungal contamination on our plates. But I think that there's certainly a potential for that. And if anyone wants to help me add some fungi to my experiments, I'm all about it.
So I'm really interested as well at how quickly, so you see the drop in diversity, basically an enrichment of these antimicrobial resistant genes in that population. Do you mean are these all plasmid borne, or are some phage delivered, or is it? I mean, because it looks like the promiscuity is really high.
Yeah, so we have one paper where we actually looked at, so again, going back to our dust samples, looking at the prevalence of mobile antibiotic resistance genes, and not all of them, but a lot of them are mobile. And we were able to actually link from our metagenomes to find those genes that were putatively mobile, and then go back to our culture collection and find isolates that had those genes. And some of them actually, the ones that we predicted would be mobile actually ended up being chromosomal. But some of them that we predicted to be mobile were actually mobile. We were unable to get them to transfer in conjugation experiments, but that, maybe we just got the conditions wrong. So I think there's a strong potential for a lot of, again, that horizontal gene transfer and the potential for mobility, especially knowing all of the genome plasticity that's out there in all of the microbial diversity. So yeah, concerning.
That's great. I mean, so there's a comment from Jack saying it'd be terrible if the antibacterial agents caused an explosion in pathogenic fungal agents.
especially given the rise in fungal infections currently. I mean, and people have heard about the Candida explosion of Candida infections and other fungal infections, and especially that a lot of those fungi are also becoming resistant to antifungals and other antimicrobials. So I think traditionally we've thought about antimicrobial resistance as a bacterial problem, but it is also a fungal problem. It is absolutely something critical that we should be looking at.
And there's a nice comment as well from Angie Sherry in the just that because some microbes produce their own pox. I mean, could these be a strategy for neutralizing environmental chemicals too? Are people looking at that?
Yeah, so there are a couple people who are looking at microbial VOCs. I think generally looking at it from the point of view of sort of human health and comfort in terms of odors, but also interesting ways of identifying microbial activity. It's obviously, when we're talking about microbial activity in the built environment, we're looking at very low biomass, very low activity. And so actually being able to detect VOCs is an interesting way of being able to look at who's actively doing something in the built environment. I think in terms of combining that work with antimicrobial, sort of looking at interactions with antimicrobial chemicals, it's going to happen. I'll say it's in progress.
So I think that concludes all the questions, Erica. I mean, it's a lovely talk, and there's some great data in there. I noticed Jack's name pop up on one of those papers as well. So yeah, so a big thanks from us, and a big virtual clap from the crowd in the background. So thank you very much for your time.
Thank you.